it's a pleasure to come back to Oxford. And I remember when I was doing my PhD and postdoc back in Imperial College, I often travel to Oxford and using the, I think it's a 950 AMR, megahertz AMR in biochemistry building. By then, it was the highest field in the world. Now, I think Broker making 1.2 gigahertz now. So, well, I just said I was UK educated. Now my group is based in China, in Xi'an, home to the Character Army. And you're welcome to have a visit. Please let me know if you go there. So in my lab, because I'm hospital-based, so I do a lot of combination of fundamental and applicational research. So we have now a phage bank of 500 phages for major pathogenic bacteria. And also we already done, well, I checked this morning, 37 registered patients, clinical trials. And also we do a lot of phage host interaction works. And also we try to develop some antibiotics from the phage taking over a strategy. So today I'm going to talk about a new work not being published related to the bacterial immunity against phages. So we all know there are two major different types of bacterial defense system. One is the most popular one is the RM restriction modification system and CRISPR. And along with that, there are a lot more being discovered. But phage always have something to fight back. So against the two popular systems, we have anti-RM and anti-CRISPR systems. So just give an introduction of like a background introduction of the two systems. So so T7 OCR is a very popular DNA mimic protein that can fight back the two very famous system, RM and the BRAC system, because this kind of protein, they mimic B form DNA and bind to the DNA recognition side of the protein that will block the recognition of bacterial protein to phage DNA. And because this kind of protein is very important, the bacteria actually develop a secondary immune system called Paris just to capture these DNA mimic proteins like T7 OCR or T4 Xi'an. So my group now is focused on the membrane-associated defense system. So there's I think by far there's only 4 systems being well characterized. So there's 2 very recent publications. One is called Kiva, one is called Zoya. And in both systems, membrane protein working as a sensor. So in the Kiva system, Kiva A is a membrane protein which can sense the bacterial attachment to the bacterial membrane, phage attachment by bacterial membrane. And Zoya system is very unique. It actually structurally resembles to the flagella motor mod AB, that membrane protein. And then they transduct a signal upon phage binding and activate a suicide reaction. And also there are some other membrane protein that can work as an effector. The gastamine and the C-bus effectors all can do that. So today I'm going to talk about our manuscript in preparation. We call a bacterial defense module, a conserved membrane-associated bacterial defense module, and its phage countermeasure. And so we call the GIMM, stand for Genome Integrity Monitoring Module. So in E. coli, we identified a potential defense module of this kind of organization, and we cloned it into E. coli. We found that it actually protects to protects E. coli against a range of phage, but not T7. Since it's a membrane protein, we first check whether the phage absorption is affected or not. Actually, we found that compared T7, T4, the absorption is not affected. And then we check the cell survival during after 10 minutes of infection, and we find that the phage, the bacteria infected by T4 was reduced greatly. That means it's a abortive infection ABI system. But if we overexpress individual modules altogether, we didn't see any growth reduction effect. That means it's probably not a TA module. It's more like a module need to be activated during certain conditions. Since it's a membrane protein, we use the live and death assay and check the membrane integrities during the infection. We found that T4 has membrane disruptions during the initial 10 minutes. So using the laser confocal microscopy, we monitor the cause of the infection or the defense. So without the gene operon, we can see the DAPI stain, which is the DNA stain. The nucleoid is gradually degrading, which is corresponding to the endonuclease released by T4 during the infection. However, with the gene module, we can see the size of the bacteria gradually shrinking and the membrane is being disrupted. So to get the mechanism of this module, so we first solve the cryo-EM structure of the GIMM-A. And the GIMM-A has no structure homology in the published database. Since it's been predicted to a DNA protein, and we check its electrostatic surface, and there is no obvious DNA binding site. However, we find that this protein binds to a double-strand DNA that's larger than 16 base pairs, but not shorter than that. So in AlphaFold3, we've actually found that this protein undergoes a very large structure rearrangement after you put DNA in there, and we check the potential interface. We found that once open up, there's quite a few positive charge side chain residues being exposed because of pore opening. And comparing these DNA bind protein structure is actually very similar to the Kiva system, the Kiva B, which is the effector of the Kiva system. That's some kind of coincidence. Based on this model, we did a mutation, mutated two positive charge residue to aniline, and we actually found that this module become toxic to the bacteria. Using confocal co-localization, we found that the GIMM-A is co-localized with the nucleoid rather than in the Kiva, which localized in the membrane. So this gave us a clue that GIMM-A is likely to be a sensor, and losing the DNA binding ability triggers the toxicity of the operon. And since after the endonuclease digested by the T4, so the genome will be disrupted, and GIMM-A will be released to the cytosol. So we solve the GIMM-AB structure. We struggle to get the transmembrane domain because very likely because it's a membrane disruptor. However, we managed to get the cytoplasmic domain and of GIMM-B and GIMM-A, and we can see the interface. The two GIMM-B dimer has a very weak interface. However, the A and B, like there's probably 12 salt bridges between them, indicating very strong interaction. However, because of the very weak interaction between the two dimers, two B dimers, we think it's probably independent from each other. Yeah, so then we mutate the key residues on the interface, and we prove that by BLI, this interaction between the mutant is greatly reduced. And this mutant, the gene operon contains this mutant, failed to protect E. coli from T4. Yeah, and also we repeated the confocal experiment, and we see that it looks like a control now. So go back to the transmembrane part of the protein, and we did a sequence alignment and found that it actually very similar to the E. coli ABC transporter MSBA. However, MSBA has 6 transmembrane domain. This one only has 3. However, there's some clues given us by the ABC transporter. So they dimerize at the transmembrane domain, and the two cytoplasma domain is relatively independent from each other. They can move freely under different conditions. So we to prove our cytoplasm domain is also kind of monomeric. So we did a SECMOS and determines absolute molecular weight at 44. So it's corresponding to a monomer. And also AMR, which you can see the signal is because AMR cannot detect a protein size of 80 kilodalton. So this is also indication of a monomer. So then we able to build a ApoB model. And then we subject this APO and bond model to the molecular dynamics simulation. So the results is shown on the right. So you can see the APO form, the membrane is nicely aligned. However, in the bond form, the bond complex induce a very large bend to the structure. We believe that's why it causes the membrane disruption. This is also the MD results suggesting the average curvature induced by the AB bond. So we then are able to propose a model that we call the GIMM. So GIMM monitors the genome integrity of the bacteria, because if the bacterial genome is degraded already, the suicide must be committed to protect the community rather than a immunity system. So then GIMM will be released to the cytosol and attached to the membrane GIMM-B. Then GIMM-B activated suicide by causing membrane disruption. So we also analyze the conservation of GIMM-A and GIMM-B. We've actually found that GIMM-A is more conserved than GIMM-B. And if we search GIMM-A itself, actually it perhaps linked to additional system like Kiva and some other unknown systems. But B is very strict linked to A in all the mobile islands. So the GIMM-B GIMM-AB operon we actually found in 24 different bacterial classes. And as I said, because the GIMM-A is a DNA binding protein and the OCR is a DNA mimic protein, it is very possible that the OCR could act as a anti-GIMM protein. So we tested it and we proved it. Well, if we knock out OCR from T7, it fails protection. If we give a secondary anti-OCR system Paris, it restored the reaction. So it's very interesting that OCR can also working as anti-GIMM protein. And last, we actually solved the GIMM-A OCR complex structure, and we show that the interface residues actually overlap with DNA binding proteins, which makes the bacteria escape the GIMM anti-gene OCR difficult because they overlap. If they don't bind to gene OCR, they will not bind to DNA anymore. So we're making them spontaneous mutation unfavorable. And that's what happened. Just quickly summarize. So GIMM is a previous uncharacterized defense system. And upon phage binding and the DNA being degraded, gene A is released from the genome and bind to gene B, and the gene B causes the membrane disruption to complete suicide, causing ABI effect, and the T7 can escape this system by producing OCR protein, and the bacterial secondary immune system pairs can restore this protection. And this work is mainly did by my PhD postdoc, Chen Huan, and also by my collaborator, Professor Yuan, who is also Oxford educated. Thank you.